Good afternoon, everybody. My name is Mohammad Sabbir, and I'm from St. Boniface Hospital Research Center, University of Manitoba, Winnipeg, Canada. I'm also associated with Also Bioscience Incorporation, San Diego, USA. First of all, I'd like to thank Cognition Therapeutics for organizing 5th International Symposium on Sigma-2 receptors. And also, I'd like to thank Dr. Susan Catalano for inviting me to share my perspectives on PGRMC1-mediated metabolic regulation. Now, as an outsider in this community of researchers, I must tell you how I got involved in PGRMC1 research. For the past 5 years, I've been doing muscarinic receptor-based research and developed an interest in signal transduction, specifically in calcium release response during muscarinic activation. And my focus was on some calcium-activated kinase. During one of my studies, I found that PGRMC1 was differentially phosphorylated in a calmodulin-dependent kinase deleted neuronal cell lines. And that was my first encounter with this protein. And then I realized that, as the name implies, it is a progesterone-binding protein, and progesterone is considered as neuroprotective. So I felt a connection between ligand-induced signal transduction and PGRMC1 signaling, which I thought could be ideal for therapeutic exploitation. Now, progesterone binds to a variety of protein, and progesterone signaling has been classified as classical or genomic, and non-classical or non-genomic. The genomic signaling occurs within hours and mediated by nuclear progesterone receptor that leads to gene expression. The non-classical signaling occurs within minutes, mediated by membrane progesterone receptor, and that leads to signal transduction, which eventually alters calcium ion concentration or cyclic AMP production. The non-classical signaling is mediated by a variety of proteins, and PGRMC1 is one of them. Now, Dr. Cahill has published several very good review papers which summarized different functional domains of PGRMC1 and their biological relevance or importance. I like to highlight some important features of PGRMC1 and some knowledge gaps which might be useful to understand the function of the role of PGRMC1 in metabolism. Now, PGRMC1 has an N-terminal domain, which is a single-pass transmembrane domain, and the C-terminal domain is a heme binding domain or cytochrome B5 binding domain. Despite the crystal structure of heme-bound PGRMC1 cytosolic domain is available, the lack of transmembrane domain structure that connects the ligand-binding domain to the intracellular signaling platform makes it difficult to understand the mechanistic aspect of PGRMC1 signaling. Therefore, more in-depth study is required in the future to understand the role of PGRMC1 as an adapter protein, acting as a signaling hub for ligand-induced metabolic regulation. Now, this structural information also highlights the limitation in developing suitable antibodies. Most of the antibodies that are commercially available are actually raised using either N-terminal peptide or C-terminal peptides, which are subjected to frequent post-translational modification, as this has been archived in Phosphosite Plus database, which is based on high throughput analysis of post-translational modification of PGRMC1 protein. Therefore, it is expected that if an antibody is raised against these epitopes, then there is possibility that depending on the post-translational status of PGRMC1, these antibodies may not recognize a particular form or the others. And this has been highlighted in my study as well as others. Now, in this slide, I have highlighted the basic signaling pathways, which is known till date, and different PGRMC1 interacting protein. As you can see that it has been proposed that heme binding to PGRMC1 causes dimerization of the protein, which then can activate EGFR signaling, leading to cancer cell proliferation or anti-apoptosis, as well as it can also initiate cytochrome binding and leads to genobiotic metabolism as well as cholesterol synthesis. In addition to that, recently epidermal growth factor receptor, insulin receptor, glucagon-like peptide 1 receptor and solid carrier family 2 member 4 receptor has been identified as PGRMC1 interacting proteins. Now, the interesting feature of this EGFR and INSR are that they are tyrosine kinases, and therefore binding of PGRMC1 to these proteins may modulate tyrosine kinase signaling, which can lead to a variety of other pathways controlling metabolic pathways. Interestingly, the glucagon-like peptide 1 receptor is a G protein coupled receptor, so therefore it is expected that binding of interaction of PGRMC1 with these receptors may involve G proteins as well as beta-arrestin recruitment. The solid carrier family 2 member 4 protein is an insulin-regulated transmembrane protein, glucose transporter protein, and interaction of PGRMC1 with this protein may influence transmembrane glucose transport. Thus, a variety of interaction of PGRMC1 with a variety of proteins, it is evident that that can lead to signal modulation. Now, in 2019, I published a paper in Journal of Steroid Biochemistry Molecular Biology that progesterone-induced Warburg effect in HEC293 cells is associated with post-translational modifications and proteasomal degradation of PGRMC1. Now, in this study, the major highlights were that progesterone PGRMC1 signaling caused a rapid induction of aerobic glycolysis in HEC293 cells, and the 70 kilodalton and 100 kilodalton PGRMC1 were found localized in the cytosol and nucleus, respectively. It was also shown that P4 caused rapid proteasomal degradation of P70 PGRMC1, which was found enriched in endoplasmic reticulum fraction. Now, PGRMC1 is a ubiquitously expressed protein. In this figure, I have shown full-length amplification of PGRMC1 in different primary human tissues, for example, adipose tissue, arterial tissue, bone marrow, cortical tissue, brain tissue, intestine, liver, smooth muscle, and the skeletal cells, skin cells. So PGRMC1 is expressed in different cell types in different tissues. But the role of PGRMC1 in a very cell type specific manner is not well understood. Interestingly, I'm showing you 2 immunoblots, and that will highlight the multiplicity of antibody recognition of PGRMC1 in different cell types. We all know that PGRMC1 is a 22 kilodalton protein, theoretically, but a higher molecular weight fraction has been previously reported by myself as well as other researchers. And in this immunoblot, you can see that a mouse monoclonal antibody, which was validated by using knockout cell lines, can recognize a P70 PGRMC1 in endothelial cells, HY926. These are all transformed cells, and MCF7 cell, MB231, which are breast cancer cells, and monocytic THP1 cells, which was derived from a lymphoma. Interestingly, when I used a rabbit polyclonal anti-PGRMC1 antibody that recognized a P25 fraction, which was highly recognized in endothelial cell but expressed to some extent less in other cell types. Interestingly, this particular polyclonal antibody recognized the P70 in THP1 cells with high affinity, but it did not recognize the P70 fraction in other cell types. So this raises the concern that a particular antibody raised using a specific epitope may not recognize all forms of PGRMC1. Therefore, some studies may not actually pick up this aspect of PGRMC1 regulation. In a recent study, which is now under review, I have shown that loss of PGRMC1 dramatically altered the endoplasmic reticulum structure and distribution of ER in the HepG2 cells. Now, in this electron micrograph, you can see that in HepG2, which is a liver hepatocyte, the mitochondria are sandwiched between rough endoplasmic reticulum. But when you knock out PGRMC1, this distribution pattern is lost, and the ER structure is also less abundant. Now, this indicates that PGRMC1 may regulate endoplasmic reticulum structure, which is very vital for mediating metabolic regulation. Interestingly, I have listed here a list of publications where molecular weight of PGRMC1 was not mentioned. And interestingly, some other publications, they mentioned the molecular weight of PGRMC1 in the immunoblots and reported a variety of molecular weights, for example, 18 kilodalton, 28, 25 to 35, or 25, 38, 50, 75, and 25, etc. Now, the reason I'm showing you is to highlight the fact that it is important to present molecular weight markers when presenting an immunoblot. And also this aspect that higher molecular fraction of PGRMC1 is cell type specifically expressed or may be subjected to recognition by using different antibodies. And previous studies have ignored this particular aspect, and this is a very important aspect to understand metabolic regulation of PGRMC1, because the higher molecular weight fractions were found associated or enriched in ER and nuclear fraction, which might have some role in metabolic regulation. In my recent paper, which is now under submission and under review, I have highlighted that cell type specifically PGRMC1 is degraded following progesterone treatment, as well as it can affect the hexokinase, which is a rate limiting enzyme of glycolysis. It can also cause differential distribution of hexokinase in different cell types. So to understand the role of PGRMC1 in metabolism, future studies must prioritize genomics. In the genomics field, PGRMC1 mediated regulation of the expression of genes that are directly associated with metabolic pathways. This can be done by using microarray-based gene expression profiling in different cell types. And the proteomics aspect of this study is first to pinpoint cell type specific post-translational modifications of PGRMC1 and its impact on cellular metabolism, and to identify the true nature of higher molecular weight forms of PGRMC1 in different primary and transformed cell types, as well as identify the involvement of G proteins and arrestin in PGRMC1 mediated signaling. In this connection, I also like to highlight one aspect of PGRMC1 expression in different mouse tissues. Now here I'm showing EGFP reporter expression in adult mouse CNS, central nervous system driven by PGRMC1 promoter in the mouse. And this data set was obtained from GeneSat Gene Expression Nervous System Atlas database. Here, EGFP reporter was inserted in the PGRMC1 gene and which was expressed under the influence of PGRMC1 promoter. So this immunohistochemistry is showing the regions of the brain where PGRMC1 is expressed, and you can see that PGRMC1 are highly expressed in the olfactory bulb, in the basal forebrain, and also in a particular some specific neurons in the dentate gyrus and other regions in the hippocampus. Interestingly, progesterone receptor is not expressed to that extent in the mouse tissue. This also highlights the importance to study PGRMC1 in neuronal signaling, particularly how it modulates metabolic activity. Also, I like to show you this data, which is also obtained from Gene Expression Nervous System Atlas, and it is showing the EGFP reporter expression in mouse peripheral nervous system driven by PGRMC1 promoter. Here, this is a cross-section of the spinal cord, and you can see the spinal cord, which is a stent very positive for PGRMC1 influenced EGFP reporter expression. And PGRMC expression is expressed mainly in the dorsal and ventral horn as well as lateral funiculus neurons, but not in the dorsal root ganglion neurons. On the other hand, you can see that progesterone receptor is expressed in the dorsal root ganglion neurons, but not to that extent in the dorsal and ventral horn neurons. So by showing this, I like to highlight that it is very important to understand the role of PGRMC1, specifically the different molecular weight fraction of PGRMC1 in neuronal signaling in different parts of the brain, particularly in a subset of neurons in the hippocampus or olfactory bulb neurons, as well as the dorsal root ganglions and the spinal cord neurons. And that might shed some novel insight into PGRMC1 mediated metabolic regulation. So again, I'd like to thank the organizer for giving me the opportunity to share these perspectives on PGRMC1 metabolic function. And I'd like to take your question if you have some. Thank you very much. And here is the contact information if you want to write to me for any collaboration or any further discussion. I'll be happy to help.